Chapter 3. Life, Diversity, and Biological Evolution. Today the theory of evolution is about as much open to doubt as the theory that the Earth goes round the Sun. Richard Dawkins. Once I read a story about a beautiful garden. In this garden lived a man named Adam and a woman named Eve. Adam and Eve shared this garden with all the creatures of the earth. Created from the dust of the ground, Adam alone was chosen by God to be his mirror image. He was given full dominion over all living things. The fish of the sea, the fowl of the air, the cattle, and every creeping thing upon the earth. In the midst of this garden lay the tree of life and the tree of the knowledge of good and evil. God commanded Adam and Eve to eat freely from any of the trees in the garden, with one exception. It was forbidden to eat from the tree of the knowledge of good and evil. Eve, who was made of a rib taken from Adam's body, was tempted by the serpent in the garden to eat the fruit of the forbidden tree. Adam also ate from the tree. Now Adam and Eve possessed the knowledge of good and evil, like God himself. God became aware of their disobedient ways and grew enraged. To show his disappointment, he cursed the serpent to crawl on its belly forever, and cursed Eve by multiplying the pain of birth. God then drove Adam and Eve from the Garden of Eden, never to return. And from these beginnings, all men and women on earth descended. I'm sure you're somewhat familiar with this tall tale. And if you're not, here is your first lesson in biblical nonsense. The above mentioned is representative of the first section of creation from the book of Genesis. In particular, I was attempting to describe God's creation of man and his role over all living things on earth, according to biblical accounts. Like any other creation story of its kind, it too shares strikingly similar features to other myths commonly found throughout human culture. Often set in a non-specific past, most creation myths describe life's earliest beginnings and humanity's role in the world. They all seem to have elements of good and evil, right and wrong. Man is usually portrayed as the centerpiece. In much the same way, Adam was created in God's image and given dominion over all the earth. Creation myths typically involve a plot and characters who are either gods, human-like figures, or animals that often speak or transform easily. This early account of Genesis fits right into that description. Indeed, even the most hardcore believer has to admit at some level that certain elements of Genesis sound rather fictional. I know many moderate Christians who lean in this direction but simply fail to admit it outright. This is almost certainly a byproduct of fear or guilt, or even ignorance perhaps. Rather than plainly rejecting it on the basis of their own logical reasoning, they accommodate these biblical inadequacies with a label of symbolism and metaphor. Unfortunately for them, the Bible does not come with a set of instructions telling its readers what is to be taken literal and what to be taken metaphorically. This is one fundamental flaw that is conveniently overlooked by believers of the Abrahamic religions. I find it quite troublesome, comprehending how anyone can possibly accept the accounts of Genesis as literal truth, especially grown adults. But on the far end of the spectrum reside those Christians who strongly support the idea that the entire Bible is a historically accurate record of actual events. In fact, according to several polls, it is estimated that up to 45% of American adults take a literal interpretation of the Genesis creation narrative. Frankly, in order to believe this, you must be willing to accept some pretty far-fetched notions. For one, according to biblical records, the Earth and universe is estimated to be between 5,000 and 10,000 years of age. Biblical scholars and theologians base these figures on the genealogies of Genesis and other dates in the Bible. On the other hand, scientifically acquired estimates tell us quite a different story about the age of the Earth. Using the latest geochronological dating methods, such as radiometric dating or chemostratigraphy, geological figures point to an approximate age of 4.54 billion years, according to the U.S. Geological Survey. When we compare biblical estimates to scientific approximates, something obvious quickly jumps at you. The disparity between the two estimates is quite wide. This is not just by a couple of thousand years, or a couple of million even, but a staggering ratio of more than 99.999999%. Well, there goes the red flag. Now ask yourself, why would the biblical God create the world just 6,000 years ago, yet make it look far older? Perhaps most crucial in this discussion is a particular theory in biology that explains the change, diversification, and distribution of living organisms over extended periods of geological time. Supported by overwhelming evidence from modern genetics, paleontology, embryology, biochemistry, and more, this explanation is the cornerstone of all modern biology. In biological circles, we refer to this as the theory of evolution. Defining biological evolution. Evolution is the change in the inherited traits of a population of organisms through successive generation. After a population splits into smaller groups, these groups evolve independently and may eventually diversify into new species. A nested hierarchy of anatomical and genetic similarities, geographical distribution of similar species, and the fossil record indicate that all organisms are descended from a common ancestor through a long series of divergent events, stretching back in a tree of life that has grown over 3.5 billion years of life on Earth. Key mechanisms in evolution. Natural selection is the gradual, non-random process by which biological traits become either more or less common in a population as a function of differential reproduction of their bearers. It is a key mechanism of biological evolution. Variation exists within all populations of organisms. This occurs partly because random mutations cause changes in the genome of an individual organism, and these mutations can be passed to offspring. Throughout the individual's lives, their genomes interact with their environments to cause variations in traits. The environment of a genome includes the molecular biology in the cell, other cells, other individuals, populations, species, as well as the abiotic environment. Individuals with certain variants of the trait may survive and reproduce more than individuals with other variants. Therefore, the population evolves. Factors that affect reproductive success are also important. Observations of the natural world suggest that living organisms have undergone modification in the past and still do so in present day. The extent of the fossil record reveals morphological similarities between organisms alive today and those that once existed. Geological records coupled with the fossilized remains of extinct species of plants and animals give us insights into evolutionary timescales, relationships, distribution, and the diversity of all living things. The genetic record of living organisms further supports the notion of relatedness and common ancestry. Species that are closely related share a higher degree of genetic similarity than species that are further apart on evolutionary scales. What appears very certain in this discussion is that living organisms were not independently created, as biblical accounts would suggest, but instead have descended and diversified over geological time from common ancestors. Genetics, complexity, and the origins of life. It should then be emphasized, biological evolution does not describe the origins of life, but rather the change, diversification, and distribution of species over geological time, and it explains the mechanisms involved in those changes. Inquiries regarding the origins of life are complicated to resolve and cross over into other disciplines of science. There is even an independent branch of studies called abiogenesis that proposes several plausible explanations for the origins of life. As mentioned in the previous chapter, some scientists speculate as to whether it could have involved the gradual chemical evolution of molecules in ocean waters near deep-sea vents. Several experiments in science have simulated early Earth conditions and tested for spontaneous chemical reactions leading to the precursors of organic compounds. The most well-known is the Miller-Urey experiment. Results from the 1953 Miller-Urey experiment generated 9 distinct amino acids from inorganic material. Recent reanalysis of the samples produced during the Miller-Urey experiment revealed that a wider range of amino acids was actually present. Amino acids are the building blocks of protein, and protein is made of the material in DNA. DNA is composed of 5 ordinary elements: hydrogen, oxygen, nitrogen, carbon, and phosphorus. A combination of these elements is bound together in strings of nitrogenous base pairs and held intact by phosphate ends to make a double helix. This is the basic structure for DNA. But the chemical substances found in DNA molecules are also present in inorganic substances. Furthermore, the chemistry for life is abundant throughout the cosmos, not limited to Earth or people or living things. The question of design now comes into play. Of course, no one will argue against the complexity of genetic structures. DNA molecules are extraordinarily intricate and complex, and unlike any molecular structure we've seen thus far. Much is often said about the complexity of the cell, and I agree. But does complexity require intelligence? For instance, a spider web is complex. Is a spider intelligent for creating a spider web? Nature produces near-perfect symmetry without external influences. Structures like crystalline, snow crystals, and self-organized convection cells look so beautifully intricate, they almost appear to have the unmistakable mark of intent. Yet, for all their beauty and variation, they simply emerge as byproducts of natural processes. Thus, complex phenomena can emerge without intelligence. I often hear creationists raise the question, can coding systems exist without design? And this brings me to my next point. Does complexity in living systems resemble a product of intelligent design? Some creation advocates would argue, yes. But this is a rule of thumb: design aims for simplicity, not complexity. For example, think of the engineers who design programming code for computer applications. One of the very basic principles of computer programming are based on the premises of simplicity and organization. Programming code that is simple and concise tends to be more efficient and reliable. Data flow relies on the precision of efficient programs. Ideally, the more simplified the code, the more efficient it may be. And the reciprocal of that is also true. Overly complex coding schema tends to be saturated, having unfavorable results in both processing and execution. Simplicity removes the unessential through thoughtful reduction. Experts say that simplicity and avoiding complexity should always be a key goal. Again, these are basic protocols of programming for computer applications. Generally speaking, there are functional benefits to designing for simplicity. One can apply the same principles to many systems and designs of engineering. It should be noted, however, simplicity is not a requirement of design, only an attribute of an efficient design. Now, looking at living systems, one cannot ignore the very obvious. Biological systems are vastly inefficient and cannot account for a great amount of waste. In fact, one might look at the results and say that biological systems exhibit characteristics that are in total contrast with intelligent design. There are countless examples of this in living systems. For one, I often hear people marvel at the machinery of the human anatomy, quite frequently giving it the label of being the perfect machine. But if this was truly the case, then why bother ourselves with doctors, physicians, or healthcare systems? For the sake of this discussion, I'll point to one particular instance because it deals directly with the issue of DNA. Cancer occurs as a byproduct of somatic mutations in the genome of a cell. Cancer can be caused by things like radiation, chemicals, infections, genetic factors, or spontaneous errors during cell division. One reliable source defines cancer as the following. Cancer is essentially a disease of mitosis. The normal checkpoints regulating mitosis are ignored or overridden by the cancer cell. Cancer begins when a single cell is transformed, or converted from a normal cell to a cancer cell. Quite simply, errors in DNA replication are the basis of malignant changes. All cancers begin in cells, the most basic unit of life. Once cancer cells get going, they can divide and grow uncontrollably, causing harm to the host organism. About 13% of all human deaths each year are cancer-related, afflicting young children and people of all ages. In light of these error-riddled missteps that occur during cell division, ask yourself once again, do characteristics such as these resemble a product of intelligent design? I believe the issue in question is not so much whether coding systems derive from intelligence, but whether poor results constitute intent or design. Moreover, biological systems appear more having been shaped and molded by evolutionary processes and trade-offs than by the implementation of a lackluster designer. I'll get into how evolutionary mechanisms shape and mold biology as we go along. So it seems that life itself lacks planning, genetic information is self-constructed in environmental context, and biological systems are vastly complex. We have also seen that nature is capable of achieving complexity on many different scales. With respect to the origin of complex organic molecules, such as protein, a 2002 Princeton University experiment concluded the following. Early and late amino acids do not correspond to a strict chronological order of introduction into the genetic code. Instead, as defined here on the basis of the changing amino acid frequencies, early amino acids are probably those which at some point lost some of their codons through codon capture and consequently became less frequent over time within proteins, whereas late amino acids are those which entered the code through codon capture, did not subsequently lose any of their codons, and therefore became more frequent over time. The present findings suggest that notwithstanding the impact of mutation over the long course of molecular evolution, with the aid of the appropriate analytical tools and insights, the sequences of contemporary proteins also provide an important avenue for exploring these early evolutionary events. New techniques in science are now allowing us to modify strands of DNA in order to see what early genomic structures may have resembled. In addition, scientists are also able to observe the effects of amino acids under early primordial conditions. This could help us determine how early organic molecules may have functioned and evolved. The role of genetic mutation in evolution. Mutations occur anywhere in the body and can be beneficial, neutral, or harmful to the host organism. As noted earlier, cancer is an example of a harmful type of genetic mutation. Other somatic mutations are so slight that they do not cause any significant harm to the host organism. Although most mutations are naturally occurring, not all mutations are necessary to biological evolution. Before proceeding, let's consider the following. First, regardless of what position you take, I think most people would agree that DNA ultimately determines the physical and behavioral attributes of a particular organism, or a group of organisms. DNA is the genetic fingerprint of an individual species, and it determines biological identity. So wouldn't major recombinant changes to DNA affect physical and behavioral appearance? Here's something more to consider. We know that mutations occur in the DNA of sexual reproductive cells. These are the types of cells that are important in sexual reproduction, and that play a vital role in heredity. Passed from one generation to the next, germline mutations may create tiny changes to the phenotype of an organism. Now ask yourself, what were to happen when tiny inheritable mutations accumulate over tens of thousands of generations? Fortunately, we have direct observations in response to that question. A recent experiment conducted by evolutionary biologists Zachary Blount and Richard Lenski of Michigan State University, put 12 genetically identical E. coli populations in complete isolation, and subjected each to various types of environmental pressures over a span of 25 years, and 50,000 E. coli generations. The result: each E. coli population developed its own evolutionary adaptations, including an increase in cell size, shape variation, growth rate, and behavior, and one population even evolved the ability to grow on citric acid. A wide array of genetic change was reported, and 4 of the 12 populations lost the ability to repair DNA. Evolution was observed in real time. Bacteria use horizontal gene transfer as one primary method for exchanging genetic information. Also, not all organisms evolve at the same rate. The rapid replication rates observed in bacteria differ substantially from reproduction patterns of larger multicellular organisms. One might observe large-scale genetic change among bacterial populations in the course of a lifetime, yet this becomes rather difficult to observe when speaking in terms of multicellular eukaryotes. This is where the fossil record helps us fill in the blanks. Nonetheless, the Blount and Lenski experiment demonstrates how environmental variables and genetic isolation can cause one genetically distinct organism to split/modify into other versions of the original. This also occurs in nature on grandeur scales. Given enough time and geographical isolation, genetically identical populations of species can deviate, split, and develop distinct features that make them look and behave different over large-scale time periods, and eventually descend into more genetically distant categories. This naturally occurring process is known as speciation. And thus, natural selection acting upon genetic mutation causes these evolutionary changes to occur. Something worth mentioning: the processes that produce new genes and functions have already been identified. One popular explanation is that genes duplicate by accident, the duplicate undergoes mutations and picks up a new function, and, if that new function is useful, the gene spreads. The experiment that identified the processes that produce new genes was carried out at the University of California. Led by Dr. John Roth, their experiment concluded the following. The cell winds up with 2 distinct genes, one providing each activity, and a new genetic function is born. Nasval, Liu, and Anderson tested this model using the bacterium Salmonella. The bacteria carried a gene involved in making the amino acid histidine that had a secondary, weak ability to contribute to the synthesis of another amino acid, tryptophan. In their study, they removed the main tryptophan synthesis gene from the bacteria and watched what happened. After growing the bacteria for 3,000 generations on a culture medium without tryptophan, they forced the bacteria to evolve a new mechanism for producing the amino acid. What emerged was a tryptophan synthesizing activity provided by a duplicated copy of the original gene. Defining speciation. Speciation is the evolutionary process by which new biological species arise. There are 4 geographic modes of speciation in nature, based on the extent to which speciating populations are geographically isolated from one another. Isolated populations undergo genotypic and/or phenotypic divergence as a. They become subjected to dissimilar selective pressures, b. They independently undergo genetic drift, c. Different mutations arise in the 2 populations. When the populations come back into contact, they have evolved such that they are reproductively isolated and are no longer capable of exchanging genes. In summary, one particular species does not simply evolve into another species, a common misunderstanding of biological evolution, but rather, splits into 2 or more genetically distinct others. Evidence from the fossil record. The most well-understood evidence in favor of evolution comes from the preserved remains of plants and animals. Fossils found throughout the planet are arranged in sequential order within horizontal sedimentary rock deposits. Sedimentary rock deposits are formed by layers of silt and mud that accumulate over geological time. Each layer of rock represents a specific time period in Earth's past. The lower layer contains older rock. The upper layers contain newer rock. The earliest fossils can be found at the lower layers, while the more recent fossils are found in the upper layers. Never has there been an instance where the fossilized remains of a particular organism appear in two distinct layers of the fossil record. Let me reiterate that point. Not one particular species has ever been found in two different layers of the fossil record. That is to say, something like the fossilized remains of early hominids have never been found side by side with fossilized dinosaurs. Dinosaurs themselves are usually found below the KT boundary, not above. Rather, we see a logically consistent distribution of species over geological time within the fossil record. As we examine each layer of rock, or strata, we see change and extinction occurring at every layer, in every time period. We find earlier forms of species that exhibit primitive traits in comparison to new ones found in later time periods. Even in precise sequence do we see the physical transition of various insects, spiders, fish, whales, birds, horses, and humans. Despite the rarity of fossilization, approximately over 250,000 fossil species have been discovered and documented. The fossil records point to clear morphological similarities between groups of plants and animals. For example, comparisons of pelvic bone structure show how modern birds are descendants of dinosaurs. The structural patterns of limb bones indicate transitions between amphibians and mammals. These limb bones can be traced back to the first amphibians that walked. All mammals today share common bone structures to their biological ancestors. Like them, we too have humerus bones, wrist bones, palm bones, and digits. Through evolutionary processes, these fundamental structures have been modified to fit different environments. In monkeys, the forelimbs became elongated to form grasping hands used for climbing and swinging from trees. A mole has a pair of short limbs for burrowing. An anteater has an enlarged third digit for tearing down ant hills and termite nests. And so on, and so on. Although the fossil record does not include every plant and animal that ever lived, it does provide substantial evidence for common descent. It also gives us a time perspective for understanding the evolution of life on Earth. Confirmation from modern genetics. In the end, no evidence for biological evolution is more tantalizing than that coming from comparative genomics and phylogenetic analysis. In many biotech laboratories, comparisons of genomic sequences are primarily used for characterizing functional regions in DNA, and identifying operations in cellular processes that help diagnose, treat, and even prevent a number of diseases that affect human beings. As it relates to evolutionary studies, comparative genomics also serves other useful purposes. For some years now, scientists have been using comparative sequence analysis to outline genetic similarities of protein functions and genetic relations between species and groups of species. Today, computer-based bioinformatics applications, such as BLAST, are used to search collaborative archives of raw genetic data. Genome databases, like GenBank, now house over 190 billion nucleotides of 260,000 organisms, including that of humans. As such, distance matrix algorithms like UPGMA, coupled with Bayesian inference, are incorporated with computational biology software that reconstruct homologous collinearity among related genomes, in order to produce phylogeny data matrices of multiple sequence alignments. Phylogenetic reconstructions depict diagrams that illustrate branching structures that look like trees, containing roots and nodes. Each node on the tree represents a taxonomic unit, and each interior node represents a common ancestral unit in which later branches diverge. By pairing sequence similarities versus that of genetically distant organisms, it is possible to produce high-degree diagrams outlining evolutionary relatedness amongst groups of species. And though internal nodes cannot be directly observed in nature, they are, however, further confirmed by morphological similarities found in the fossil record, in direct comparison to that of modern species, suggesting evolutionary processes at work. While DNA-DNA comparisons determine genetic relationships, molecular clocks determine the extent of those genetic relationships over a given period of time. Molecular clocks use the rates of molecular change to deduce the time in geological history when two species diverged. In terms of evolutionary biology, molecular clocks are used to estimate the time of occurrence of speciation. Time estimates are represented in phylogeny by the length edge of each node, which infer the rates of origination, extinction, and patterns of diversification. Embedded within the phylogenetic tree of humans and other great apes are unmistakable marks of African origin. Detailed analysis based on DNA-DNA hybridization reveal close genetic ties to chimpanzees, gorillas, orangutans, and gibbons, and it suggests divergence with those lineages and common ancestors sometime in the past. Human DNA sequences are approximately 1.8% divergent from those of our nearest genetic relative, the chimpanzee, and 2.3% from gorillas. Additionally, humans and chimps share 7 different instances of virogenes, while all other primates share retroviruses congruent with resulting phylogeny. Direct evidence for common lineages between humans and other members of the great ape family is also found in human chromosome 2. For instance, humans have 23 pairs of chromosomes, while chimps, gorillas, and orangutans have 24. Chimpanzees have near-identical DNA sequences to human chromosome 2, but they are found in 2 separate chromosomes. The same is true of the more distant gorilla and orangutan. It is thought that human chromosome 2 is a result of an end-to-end fusion of 2 ancestral chromosomes that resulted in the correspondence of chromosome 2. Or in other words, humans have 1 fewer chromosome pair in their cells than apes, due to a mutation found in chromosome number 2 that caused 2 chromosomes to fuse into 1. The fusion between 2 ancestral chromosomes therefore presents very strong evidence in favor of a common ancestor between humans and other members of the great ape family. Analysis of human DNA reveal even more startling insights into our regional beginnings, and our relationship to one another. In 2003, scientists at the Human Genome Project announced that they had mapped the entire human genome. This was a significant point in modern science. For the first time, scientists were able to reconstruct a phylogeny based on the genomic data of different human populations. Of the 3 billion base pairs in the human genome, one particular type has significance in determining relatedness among immediate populations. Mitochondrial DNA, or mtDNA. Mitochondrial DNA is inherited solely from the mother. This form of DNA is found to mutate more rapidly than other molecules. Mitochondrial DNA is thought not to be affected by the process of natural selection, allowing mutations to accumulate more freely in non-coding DNA. Using the latest techniques in bioinformatics, scientists have sequenced thousands of base pairs of non-coding regions in human mtDNA. Several thousand human volunteers, representing distinct populations from all parts of the world, including a number of different tribal groups in Africa, were subjected to genetic testing. In almost all cases, 2 or more people sharing the same sequence also belonged to the same population. Of the 100 different sequences identified, the greatest diversity of sequences was found among the various African populations. The data would later reveal a human family tree by which all present-day human sequences could be narrowed down to a single ancestral sequence. Comparative sequence analysis of human populations indicate a point of origin. East Africa, about 120,000 years ago. Darwin was right. There is an overwhelming consensus within the scientific community. In fact, it's hardly a debate. According to a recent Gallup poll, over 95% of all world scientists accept evolution as the most valid set of principles that best explains the diversity of life. One news article published in Newsweek said the following: by one count there are some 700 scientists with respectable academic credentials out of a total of 480,000 U.S. Earth and life scientists who give credence to the counter-evolutionary argument, also known as creation science. Something worth noting. Belief in creationism is inversely correlated to education. Of those with postgraduate degrees in the United States, 74% accept evolution as a valid explanation, rather than the alternative claim brought forth by critics of evolutionary theory. It is important to point this out. Misconceptions are prevalent, most of it being the product of lies fueled by Christian elements and other creation advocate groups. Furthermore, I cannot presume to call myself an educator and just stand by and do nothing. Let me make this clear. All independent disciplines of science converge on this issue. Dr. Richard Dawkins said it best. Today the theory of evolution is about as much open to doubt as the theory that the Earth goes round the Sun. So, where does that leave us? I'd say it's pretty clear. Charles Darwin was right about common descent, despite what you might have heard. Naturally, the repercussions stemming from evolutionary studies stretch far beyond scientific circles and into congregations, churches, and mosques. Biological evolution all but exposes the illegitimate claim of divine creation. Darwin himself understood this too well. He knew that while his theory of evolution, powered by the mechanism of natural selection, produced a good explanation of adaptation, it also removes the need for intelligent design. And thus, species of living organisms were not independently created, but have descended and diversified over time from common ancestors. To date, no other scientific theory so elegantly explains this. Evolutionary theory has withstood the test of time, by way of vicarious experimentation, observation, analysis, and relentless criticism, though opposing viewpoints still cling to the concept of intelligent design. As a person of the biological sciences, I cannot subscribe to such misguided notions that suggest static biological states. Proper examination of the natural world reveal evolutionary trajectories, some random, others non-random, and all having observable genetic implications. It is only when we apply evolutionary explanations to living systems that it becomes ever so clear. The world was not specifically designed with us in mind, but rather we long since adapted and conformed to our surroundings, only giving it the illusionary appearance of design.